Okay, we're going to be looking at stem cells and differentiation as part of cell specialization. So cell specialization is a process where undifferentiated stem cells develop into highly specialized cells with specific functions and properties within the body. And the characteristics of stem cells is that they can repeatedly divide in two, and they can either stay undifferentiated with no specialized function but retain the ability to keep on differentiating, or they can differentiate into cells with different specialties and then stay within that fixed shape and function. And stem cell niches are kind of where stem cells live. They're locations of stem cells within tissue that you can, for medical purposes, take stem cells from if you wanted to then differentiate these cells for therapeutic uses within the body. And in an adult's body, the stem cell niches are 1 in your bone marrow and 2 in your hair follicles. So these are stem cells that are repeatedly regenerating to help with blood production as well as hair regeneration. So the stages of differentiation from a stem cell starts off with the zygote, which is also known as totipotent. So the totipotent stage is where the stem cell can develop into any sort of cell, and this includes the placenta, and but only the zygote can develop into the placental cell, and this is when the cells are still within utero. Then we have pluripotent embryonic cells, and pluripotent cells can also develop into any specialized cell type except for that of the placenta. So the separation between the zygote and the pluripotent cell is that only totipotent cells can differentiate into placenta cells, whereas pluripotent cells can develop into any other specialized cell. Then, as it develops into a multipotent cell, it then becomes fixed and it retains that fixed shape and structure. And then if it were to go and differentiate again, it would only be into related cell types. So if you had a undifferentiated stem cell and it differentiated it into a muscle cell, it could only then multiply itself into another muscle cell and not a undifferentiated cell. So yeah, those are the 3 levels of organization, from totipotent to pluripotent to multipotent. And the reason this is important is because specialization allows cells to gain properties that aid the entire system to function and perform different types of specialties. And this is done through selective gene expression, where your DNA code produces different sets of proteins, but in order to produce sets of proteins that actually have specialized functions, these are gene expressions that are selective. So you take certain types of the code and then you mute the other ones, so you come out with unique protein codes that can code for exactly what you're looking for and allow your cells to specialize. Then we have signaling chemicals, also known as morphogens. These are really cool because when a baby is in utero, you actually don't know which end of the body will develop into which organs without the release of signaling chemicals. So in the context of an embryo, we have retinoic acid as our morphogen, and basically as the retinoic acid spreads through the womb and through the uterus, the amount with the highest retinoic acid, which is the chemical gradient, develops into the colon, and as the chemical gradient becomes more diluted, it's assigned for the different parts of the organs to develop, and it kind of just indicates which direction for the embryonic womb to develop in for morphogen gradients. Yeah. And then, yeah, that is everything for cell specialization without talking about adaptation.